Hello, everyone. In the previous lecture, the cell membrane, we talked about the properties of the cell membrane. And one thing I hope you took home is how these phospholipid bilayer with proteins in it and so on represent not only a barrier for the cell to protect itself, its inner contact, but represents also a big hydrophobic barrier for the movement of material and for the connection of the cells with the outer world. So that's exactly what we're going to be talking about in this lecture. We're going to be talking about two important things. How does a cell deal with the movement of material across this big phospholipid bilayer, this big hydrophobic barrier that defines the inside and the outside of the cell? And together with that, we'll talk about at the end also how do cells communicate with each other, and most importantly, how do they transfer that information, that communication from the outside into the inside of the cell so that the cell can perform a specific task based also on signaling from other cells that can be far away in the body as well. So let's start talking about the transfer of material across the membrane. Now, material can move in different ways, depends on the size and the overall properties in terms of polarity and so on of the molecule. So there are two main ways in which molecules can move without the need of energy. In this case, we'll talk about passive type of transports. There are different types. We'll go through them. So there is simple or facilitated diffusion. We'll see what it is. And there is basically a similar movement, but specific for water, which is referred to as osmosis. All of these type of movements, simple and facilitated diffusion, as well as osmosis, do not require energy, and they're passive type of transports. Then there is a second group of movements. Sometimes molecules have to be moved against their concentration. And so cells need to invest energy to do that, and they perform what we call active transport. Or if it comes to large amounts of molecules, or large molecule, really big, or large complex of molecules, such as bacteria, viruses, then they need to use membranes, and they perform what we call exocytosis or endocytosis, depending on the direction of the movement. So in this case, again, active transport, exocytosis, and endocytosis, they all require energy, and they're basically active type of transports. Starting with passive transport. So, or first of all, fusion, whether it's simple or facilitated. What we mean by diffusion, we mean the movement of molecules following their concentration. Now, we have to understand one thing. So we are dealing with a big hydrophobic barrier, which basically is almost impermeable to most of the biological molecules. If you recall, carbohydrates, simple carbohydrates, proteins, amino acids, stuff like that, they're all polar molecules. They have a very hard time going through a hydrophobic environment such as the membrane. However, there are some tiny small molecules, which can be like oxygen, carbon dioxide, some small non-polar molecules that can still manage to go through this barrier, freely diffuse across the membrane. Okay, so some of them do. Not all molecules do need the help of a transporter or something else. So small non-charged molecules, again, such as small gases, water, and some small non-polar molecules can actually freely move across this membrane. Now, how do they do that? They do that by doing a movement referred to as diffusion. Diffusion is the movement of molecules following their concentration gradients. What do we mean by concentration gradients? We mean that difference in concentration between a point A and a point B in a solution. And this point A and B could be also the outside or the inside of a cell membrane. So if there is a difference in concentration of a certain molecule, imagine like glucose, for example, in one point of a solution versus another one, glucose molecule will move both directions in reality, but they would have a net movement going from the high concentration to the low concentration. This movement we refer to as diffusion of glucose in this specific case. It's exactly what is happening here. You can see it here. For example, if you put a drop of a dye in the water, you are creating now a point A here where this dye is highly concentrated, much more concentrated than in the remaining of the aqueous solution here, which will be point B. And therefore, these molecules are moving from A to B, B to A, but the net movement will be from A to the rest of the solution, and therefore leading to the even of the concentration throughout the solution. Movement does not require energy. It follows the second law of thermodynamics. We'll talk about that in the next lecture. And most specifically, they're driven by entropy. Just take my word for it. We'll talk about that in the next lecture about that. And now, these obviously will last until there is a difference in concentration. As soon as a difference in concentration is done, the movement, the net movement will stop. The movement in both directions will still take place, but the net movement, so the movement left to right, right to left, will be equal to each other, and therefore there will be an equilibrium reached. Okay. You can imagine how diffusion can be affected by different parameters. First of all, diffusion for each molecule is independent from the diffusion of the other molecules. So if you have glucose and sucrose here, both of them would follow their own concentration gradient. Second thing is there might be different parameters, the amount of energy. So the heat in this solution will make it go faster or slower. Increase the heat usually goes faster. And the difference in concentration. How big is this concentration gradient would affect how fast this movement would take place. If it's very big, it will go faster, it will probably slow down, than if a small difference in concentration. Again, diffusion is any movement of molecules following their concentration gradient and does not require energy. Now, small molecules, such as small gases, you remember small non-polar molecules, non-charged molecules overall, they can move across the cell membrane on their own. In this case, if they don't need any help, they just move freely across this hydrophobic layer, you talk about simple diffusion. Still following the concentration gradient from point A here, high concentration, to the point B where there is low concentration. Some molecules are too big to do that on their own, but they can still do it using passive transporter, which are proteins that are embedded within the cell membrane. And basically what they do, they create a small polar environment here for these molecules to actually move through. Since they cannot move through the cell membrane, which is hydrophobic, they can, however, move through the transporter to this polar opening here created by the transporter following their concentration gradient. In this case, this is referred to as facilitated diffusion. Both of them are diffusion. They do not require energy. The molecules move through the concentration gradient, but in one case they do it on their own. In the other case, they do it with the help of a passive transporter. Okay. Now, this is necessary for molecules, for example, glucose. They have their own transporter here, which increase the movement by 50,000 times, right? So it speeds it up very, very significantly compared to the movement of glucose on. Now, this is just resuming what we just said. So both of them will follow concentration gradient. There is simple diffusion. There is no transporter involved. This applies only to small molecules. So small gases, small nonpolar molecules. Facilitated diffusion applies to larger molecules. They would have their own specific transporter. So glucose has the glucose transporter, cannot use the fructose transporter. And in this case, they will move, again, following the concentration gradient, but with the help of the passive transporter. This is the same one, duplicated slide. Now, depending the passive transporter, so those proteins that help with the diffusion of molecules, they can be of two types. They can be referred to as channeled proteins, which do not bind the molecule. They usually transfer ions channel proteins, or water, for example, and carrier proteins. In this case, the carrier proteins, in order to move the molecule, they have to bind it, and it's the binding of the molecule that causes the opening of the protein, allowing the diffusion from the area of high concentration to the area of low concentration. Let's go through some example here. So aquaporin, for example, is a passive transporter that allows the movement of water from the area of high concentration, area of low concentration, if you want, of water in this case. And so, and what it does, all it does, it has this opening. It doesn't really bind water, but it creates this polar opening specific for water, which allows it to go from where the water, there are more water molecules per unit versus where there are less molecule of water per unit. So where the concentration of water will be lower. Other examples are ion channel proteins. These can be closed at times, but they don't open because of binding ions, but usually they're opened by voltage changes. You see, when we talk about the actual potential in neurons through the axon, how it's generated, we'll talk about it. So in muscle cells. So there are channel proteins or ion channels, so potassium, that moves sodium and so on. They get activated when the voltage in the cell membrane changes to a certain level. They're normally closed, so nothing will move, but with a change in voltage, they open up and the ion, the specific ion, can move. It's not the binding of the ion that opens it. So they're referred to as channel proteins. Instead, carrier proteins, they do require the binding of the molecule in order to open. And so here you see an example. This could be the glucose, for example, transporter here, where you have that in the normal configuration, it will be open to the side where the higher concentration of glucose is. So this is the concentration gradient, higher on this side, lower on this side. So the molecules will move from the high concentration to the low concentration by diffusion. And now what happens is that there is a binding site for this molecule. Again, imagine this glucose. It will bind here, and it's the binding of the molecule to the transporter that causes a change in configuration. So imagine these alpha helixes, whatever is making up this protein, changing their tertiary structure or quaternary structure if it's multiple units. And this basically leads to the change in configuration and the change of the orientation of the binding site. Now, it's not oriented on this side anymore, but the binding site is oriented on this side. And this causes the release of the molecule per se. So as a result, with this cycle, one molecule would have been transferred from the area of high concentration to an area of low concentration, following the concentration gradient, therefore passive. So this will be a passive facilitated diffusion. And then once the molecule is released, now the transporter goes back to the original configuration. There is no binding there anymore, changing the configuration. It goes back to the original configuration and therefore can bind another molecule and repeat this cycle over and over, again, causing the movement of many molecules in this direction, following the concentration gradient by facilitated diffusion. Second type of passive transport, say before, it's osmosis. Osmosis, if you want, is very similar to diffusion. The only thing, instead of involving the solute, so glucose will be dissolved in water, for example, or potassium will be dissolved in water, and that's referred to as the solute, okay? Now, and the solvent will be water, but sometimes you're in situations where the solids cannot really move. Okay, you may have semi-permeable membranes that are not able to transfer these molecules. This could be a combination of different molecules. Now, this is a problem for living organisms. When you have a lot of solutes accumulated on one side versus the other one, there is a pressure that is exerted on this membrane here, which will eventually cause it to break. So how is this compensated? Is by moving, instead of moving the solute, which wouldn't have any transporter or anything, or the capacity to go through the phospholipid bilayer, this is compensated by the movement instead of water. So At the beginning, you would have that there is a lower concentration of in this solution, higher concentration here. But now this concentration is evened out not by moving the solute, but water. You would have that water will be moving from the area of low concentration of solute to the area of high concentration of the solute. And this will keep going. This is referred to as osmosis, the movement of water. This will go on until the concentration on the two sides will be evened out. Most of the time, this concentration, relative concentration, when it comes to osmosis, is referred to as tonicity. So again, tonicity is the expression of concentration of two solutions, like this one here on the left, compared to the other one. Not in molarity on specific units, but compared to each other. So the solution on the left is less concentration than the one on the right. Therefore, this is referred to as hypotonic compared to the other solution. The other solution is the opposite, is more concentrated, and therefore is represented or referred to as hypertonic compared to the one on the left. Now, the movement of water takes always place from the hypotonic solution to the hypertonic solution. And obviously, this is the reason behind this is that you have to have, this has to happen so that the two concentrations can get to the same, become what we call isotonic, okay? So osmosis is another type of passive transport happening in living organisms, not only in living organisms, that leads, again, in this case, moves water from one side to the other one, from the area of low concentration of the solute, so hypotonic area, to the higher concentration, the hypertonic solution. This is what you see in different scenarios. For example, if you take, you can do an experiment. So if you take a certain concentration, a molecule like sucrose, 2 molar in this case, and you put it inside a semi-permeable membrane. So this membrane here will be permeable to water, but not to the solute, so in this case, sucrose. So sucrose cannot move out. Now, if you put it in a solution, which is pure water, you will find that solution, the pure water solution will be hypotonic compared to the solution inside of the balloon. And therefore, you would have that the water will be moving through the semi-permeable membrane from the area of low concentration to the area of high concentration, from the hypotonic solution to the hypertonic solution, and therefore will cause the balloon to swell. You would see by seeing the balloon swelling. The opposite would happen if you put in a solution that has a higher concentration of solute, 10 molar sucrose. In this case, the solution inside the balloon is the hypotonic solution. The water will be moving out, and therefore you would see the balloon shrinking. If instead you are in isotonic conditions, so the solution outside has the same concentration as the solution inside, you have net movement of water in both directions but equal. So you would have no net movement of water, no osmosis happening, and the balloon would keep its own original shape. This you can see even in cells. These are red blood cells. If you do the same thing, you put them in water, for example, they would actually, so this is the natural form. They look like donuts, okay? That's their natural shape. If you put them in a hypotonic solution, the solution they're in would tend to enter, the water would tend to enter inside the red blood cells, getting them to swell to the point where they would actually explode and release their content. The opposite, put them in a solution that is hypertonic, a higher overall concentration of solute compared to the inside of the red blood cells. In this case, the water will move from the inside of the cell to the outside, and they will shrink. Instead, if you put them in an isotonic solution, the movement in and out is the same. There is no real osmosis happening. The cell will keep their own original shape. Now let's move from the passive transport to the active transport. So there are two types, as I mentioned before. There are those that deal with the movement of single molecule, one at a time, what we call transporter. And then there are those that deal with the movement of bulk material, so lots of molecules at a time, at each time, or large molecules, such as even complexes of molecules like bacteria viruses. In that case, we talk about exocytosis, endocytosis, or vesicle-mediated transport. In both cases, this movement move the material in opposite directions, and therefore to the concentration gradient, and therefore require energy. Let's start with the active transport. Now, sometimes understand some molecules need to be moved against their concentration gradient. They cannot always move by diffusion. For example, you need to remove waste. Okay, waste tends to accumulate inside a cell. If you have already a high concentration of waste outside, now the waste, if you had to move by diffusion, it would not move, right? If anything, it will move backward inside. But now, so you still want to keep removing this waste, which could be toxic to the cell. So the cell have to basically pump this waste outside. And they do that using specific transporter that do, at this point, require energy to move this waste in this example. But this could be other molecules. There might be already a high concentration inside the cell, but you still need to get more to keep going. And so you still have to move them against the concentration gradient. Again, you would have some transporter, active transporter, doing that using energy. You need energy because this movement is going against the second law of thermodynamics, right? Is going against entropy. And therefore, you have to add energy for this to happen, to keep going. This deal, again, as I say, with removal of waste, movement of nutrients, or movement of ions, for example. You have to keep concentration of ion on the two sides. And so you have to, so for example, you see when we talk about neurons, muscle cells, they have to keep this differential, and they have to do it using active transport. Now, there are two types of active transporter or active transport in general, and both of them require energy. The difference between them is how they get the energy. So in primary active transporter, the energy is given to the same transporter by a molecule of ATP. Whereas in secondary active transporter actually does two types of movements. It does the active transport of a certain molecule, and it fuels that not by using ATP directly, but by dissipating the concentration gradient of another molecule, so doing facilitated diffusion of another molecule. Facilitated diffusion follows the second law of thermodynamics and therefore releases energy in the process. And that energy can be used to do the other movement, the active movement. In the end, you see that even in this case, though, the energy is coming from ATP through a second transporter, which is involved in the full process. Let's look at two examples. Primary active transporter. So here you see some ion pumps. Again, ions need to be kept at specific concentrations across the membrane in some cells, like muscle cells, neurons, and so on. And so you can't let them diffuse inside the cell all the time. So you have to keep that, and to do that, you have to pump them out. Here you have an example of some ions there. So in this case, this ion would have a high concentration on this side versus this side. So the concentration gradient is going in this direction. However, you want to keep this. If the ion diffuses in, the cell would not be working. So you need to keep pumping ions against the concentration gradient. How is this done? It's done by transporter, an active transporter. In this case, will be a primary active transporter. Then what it does, it binds the ion, and then at the same time or right before, it gets the energy to change the conformation by binding to a phosphate group coming from a molecule of ATP. So when you break ATP, you remove one phosphate. In reality, the energy that was in the bond between this phosphate and the other two phosphate here doesn't really get, but it gets transferred. So this binding here transfers the energy to this transporter here, this protein. And these energies, that's what causes ultimately the change in conformation. The change in conformation, what it does is now it reorients the binding site from the ion from the area of low concentration to the area of high concentration, so that the ion can be released on the area of high concentration. When that has taken place, now the change in conformation that happens here causes the release of the phosphate group here, and therefore the active transporter, this primary active transporter, goes back to the original conformation, getting ready to bind another ion but together with ATP and moving another ion against the concentration gradient. Again, in this case, the energy is coming straight from ATP that binds to the same transporter. So it's a primary active transporter and primary active transport, the movement itself. You have a very specific case that you see when we talk about neurotransmission, for example, is you have a sodium-potassium pump, which is essential to keep the concentration of both sodium and potassium ions across the membrane in the axon, for example. So how does it work? Well, this pumps both sodium and potassium against the concentration gradient. So this is the outside of the cell. The sodium is at high concentration outside of the cell and a low concentration inside. And it's the opposite for potassium. And both of these gradients have to be kept intact by these cells so that it can be ready to accept another and generate another action potential later on. We'll talk about that in BIO359. So how does it work? Well, in the native configuration, when ATP is not bound there, it has three active binding sites for sodium ions. So it can bind 3 sodium ions in the area of low concentration for sodium. Now, the binding of ATP with a transfer of a phosphate group to these transporter now causes a change in conformation, which basically gets these 3 binding sites exposed to the side where the sodium concentration is a higher concentration. This causes the release of the sodium ions in the area of higher concentration. So the active pumping of sodium, so the active transport of sodium has been achieved in this case. Now, the same pump, though, has also 2 binding sites for potassium here. And on the outside, that's where you have the low concentration of potassium, which then can bind into these binding sites. When this happens, now the phosphate groups gets released, and this pump, this active transporter at this point, once it loses the potassium, the phosphate, attached to it, it goes back to its original conformation. By doing that, now the 2 binding sites for potassium are actually exposed to the area where the potassium concentration is higher. The 2 potassium ion gets released. By doing that, you have achieved also, with just one cycle, the movement of potassium ions, 2 potassium ions, from an area of low concentration to an area of high concentration. So the active transport of potassium has been achieved as well. So in one cycle of this pump, you can achieve with a single molecule of ATP the active pumping of 3 ions of sodium from an area from the inside to the outside, and the opposite movement of 2 potassium ions from the outside into the inside of the cell. Again, this is a primary active transporter because it binds directly to the phosphate group, so it uses ATP directly to do this movement. Secondary active transporter instead do not bind ATP, they don't use it directly, but they do basically 2 types of movements at the same time. One is the active transport of the specific molecule. The second is they do a facilitated diffusion of another molecule. And they use the energy from that facilitated diffusion. Again, facilitated diffusion goes from an area of high concentration to an area of low concentration. This follows the second law of thermodynamics and causes the release of energy, which can be used for something else. In these active transporters, they use that energy to pump the other molecule against the concentration gradient. Now, this can happen in 2 directions. So the molecule that is doing the facilitated diffusion goes in the same direction that the molecule that is actively transported. In this case, you would talk about symport right here. They're going on the same direction. You refer to this as symport. This will be a symporter, the transporters. Or they can go in opposite direction. So, and in this case, you will refer to this as antiport movement, and this molecule, this transporter, will be referred to as importer. Okay. Both cases, there is energy involved. But in this case, instead of coming directly from ATP, it's coming from the facilitated diffusion of a different molecule, which usually is an ion. Okay. So now let's see an example here. You have it here on the right side. So for example, glucose is actively transported against its concentration gradient. So from an area of low concentration to an area of high concentration using a importer. This importer uses the energy that comes from the facilitated diffusion of sodium from the outside, where you remember it's at high concentration, to the inside. So it has facilitated diffusion of sodium from the area of high concentration to the area of low concentration. So this releases energy, which then is used to pump glucose at the same time against its concentration gradient. Now, notice one thing. In this case, this is a secondary transporter because it doesn't bind ATP directly here. The energy comes from this concentration gradient dissipated relative to the sodium ion. However, if you think, as we saw before, the concentration gradient of sodium between the two sides is kept at a sodium-potassium pump, which uses ATP directly. So in this case too, indirectly, the energy here is coming, if you want, from ATP through another pump, but it's still coming from ATP. The only difference is that it doesn't come directly, so it's referred to this movement here as a secondary active transfer. Let's move on to the next level, final movement of material across the membrane. Sometimes cells have to move not one molecule at a time. It would take too long to move them. They want to move large amounts of molecules quickly. Or they have to move big complexes in, bacteria, viruses. And so instead of using transporters, which are proteins that have a limited size that they can carry, what they do, or limited number of molecules per unit of time that they can carry, they use membranes. They form vesicles. And now this movement, depending on the orientation, is referred to as exocytosis or endocytosis. It requires energy in this case as well. So it's an active type of movement. So when it comes to exocytosis, now you have formation of vesicles coming, for example, from the Golgi. You remember? The Golgi would pack molecules inside some vesicles, and these vesicles will be directed to the plasma membrane. They will fuse plasma membrane and they will release their content. By doing that, you would have achieved the movement of material across the cell membrane, in this case by packing it inside these exocytotic vesicles. Whereas the opposite happens when you talk about endocytosis. In this case, the material is taken from the outside into the cell by forming vesicles at the plasma membrane level. The material gets trapped in a vesicle, endocytotic vesicle is formed, and this material gets trapped inside. Now, these vesicles formed here and here, usually they come from recycled material. There is a cycle of these vesicles taking place there. So, as we said before, when we talk about exocytosis, this refers to the release of molecules to the outside of the cell. Exo means outside. It's done through secretory vesicles. For example, your B cells, they pack antibodies here, forming secretory vesicles, secretory vesicles, which then they will fuse with the cell membrane, and by doing that, they will release the content of the secretory vesicles to the outside, in this case, antibodies, for example. Now, again, these movements require energies, as we say before, and is very specific. Okay, it's not a random movement. It's directed through the presence of proteins on the outside of the vesicles that direct the movement of these vesicles specifically to the plasma membrane. The opposite is when you have that the material from the outside is taken in through the formation of vesicles. This is referred to as endocytosis. Now, there are different types of endocytosis. Okay, they work in different ways. Some of them are specific, some are not. There are three main types that lead to the formation of what we call endocytic vesicles. the vesicle formed through endocytosis. The three types are bulk phase endocytosis, which is called usually pinocytosis. There's a receptor-mediated endocytosis, and finally phagocytosis. Now, of this movement, the first one, pinocytosis, is non-specific, meaning that it takes material in all the time, keeps forming these vesicles all the time. No matter what is the material outside, it will take it in. So it takes, is referred to as pinocytosis, we'll see in the next slide, because it takes drops of water with whatever it's inside there. Whereas the other two movements, receptor-mediated endocytosis and phagocytosis, they're all mediated in the end through the specific recognition of whatever it's outside. So the formation of the vesicle takes place only when that specific material is detected through receptors and therefore is brought in ways, but is specific type of movement. Pinocytosis, again, this is referred to as bulk phase endocytosis. There is nothing that triggers it. So some cells, for example, amoeba, put a dye outside of an amoeba, will quickly move inside the amoeba because amoeba keep doing pinocytosis. They keep ingesting all the material outside, forming these endocytic vesicles. And all they do, they form invaginations. Okay, so in this case, in pinocytosis, the same thing for receptor-mediated endocytosis, the membrane bulges towards the inside of the cell. So that's referred to as an invagination. And this takes place until the membrane fuse and the endocytic vesicle is formed. This endocytic vesicle then will be taken by, will fuse with a lysosome. Do you remember, right? Lysosomes are these organelles inside the cell packed with hydrolytic enzymes, or enzymes that can digest almost anything. Okay, so different enzymes, each enzyme can digest different type of molecules, but combined together, they can digest pretty much anything. And so they will be fused with this vesicle, the enzyme will be released, and they will start digesting all the milk taken in. Again, this is not specific. It happens all the time. It's not triggered by the specific molecule outside. Instead, there is a second type of movement referred to as receptor-mediated endocytosis, which instead is triggered by the binding of specific molecules to a specific receptors. Cells do have, and let's move directly here, they do have on their outside specific receptors that can be used for different things. In some case, they can be used to detect the presence of specific molecules that need to be brought in. So when the molecules are present outside, they will bind to the specific receptors, which then will fuse or tend to cluster together, also with the help of this protein under the membrane, referred to as clathrin, named clathrin. And this will cause ultimately the invagination of the membrane, as in pinocytosis. You can see it here as well. This is clathrin. These are the molecules bound to the receptor. And the formation of a vesicle. So by doing that, basically in this case, you would have the formation of this vesicle, which is triggered by the presence of a specific molecule. So it's a specific type of endocytosis happening. And so as a result, this molecule will be brought in through the formation of endocytic vesicles, which will be first coated with clathrin. Now these endocytic vesicles would then fuse again with lysosomes, release their content, and these vesicle here with the receptor, once this is released, these molecules bound here are released to the lysosome, they would actually be reused, or they will fuse again with a cell membrane that will be recycled. Okay. Again, in this case, receptor-mediated endocytosis is a specific type of endocytosis. It's triggered through the binding of specific molecules to their specific receptor, which recognizes only these molecules, and this leads to the formation, the invagination of the membrane through the help of clathrin here, formation of vesicles, which then fuse with lysosomes, and the vesicles get recycled there. Last type of endocytosis is phagocytosis. Now, these deals with large molecules, okay, large objects such as bacteria, different cell parts, viruses are taken in through phagocytosis. In this case as well, this is a specific type of endocytosis is triggered through the recognition of some antigens in the bacteria or viruses. So the cell membrane will sense the presence of, let's say, a bacterium in this example through binding to specific molecules present here, referred to as antigen, through specific receptors here. And this will cause not the invagination of the membrane in this case, but the formation of the membrane outward, the extension outward of the membrane. Okay, so the vesicle in this case, the endocytic vesicle, is formed by surrounding the object instead of bringing it in for invagination. So it gets surrounded by the membrane until the membrane fuses at the opposite end here, forming the endocytic vesicle here, which then would fuse with the lysosomes and would cause the digestion of the material brought in. Bacteria, viruses, will be digested inside the cell. This also is used to then recycle and the antigens in some cells and present it to the immune system. We'll talk about that in BIO359. Okay, so the three types of endocytosis we talked about. We talked about pinocytosis, completely non-specific, happens all the time through invagination of the cell membrane. Receptor-mediated endocytosis and phagocytosis instead are specific. They're triggered through the binding of specific molecules, whether they're free or attached to a bacterium or a virus. And this cause in receptor-mediated endocytosis, that cause the invagination of the membrane. In phagocytosis causes the expansion of the membrane outward. Leads to the formation of an endocytic vesicle, which then is fused with the lysosomes. Now, all of this movement, pinocytosis, phagocytosis, and receptor-mediated endocytosis, they all require use of quite a bit of ATP, of energy, in order to proceed. Last thing we talk about in this lecture, so we are done with the transport. We'll summarize it at the end in the summary slide. Now, last thing we were going to talk about is how do cells overcome the presence of this big hydrophobic barrier, which is the cell membrane, to receive signals that is coming from other parts, from the outside of the cell. Cells that do communicate with each other all the time. You may have communication that takes place at long distances. For example, in your body, you have your hypophysis that communicates with your kidney cells, the smooth cells in your uterus, if you have one. And they do that using, for example, short peptides, like short sequences of amino acids, that then carry some specific information. How is this information, for example, transferred from the outside of a cell, let's say in your uterus or in your kidney, to the inside, and how does it cause a specific response? How is this happening? All of this process is referred to as cell signaling. Now, when it comes to the cell membrane, this transfer of information, whether is this short peptide or something else, a hormone, that's actually usually a hormone that goes straight in. So how is this information transferred from the outside to the inside so that there is a specific effect, a specific response? It's done in three easy payments, in three steps. First, there is the process of reception. There is in every membrane, there are molecules called receptors that can recognize specific signaling molecules. Okay. Once they bind to that, they transfer that signal from the outside of the membrane to the inside, where they cause the second step, which is referred to as transduction. Transduction now happens when the receptors start triggering a series of reactions, which are referred to as a signaling cascade, which ultimately affect the final molecule, which is the effector, which triggers what is called the response. So three steps. The signal is received through a receptor, is then transduced inside of the cell and amplified through signaling cascade. And then these ultimately reaches the final effector molecule, which leads to the response. The response could be, for example, a cell start producing antibodies, or producing more antibody, or a cell start dividing. That's what we mean by response in there. So talk about receptor first of all. So these are those molecules that do recognize the signal coming from outside. So imagine these red molecule being, for example, a short polypeptide, oxytocin. This will be your cell in these muscle cells in your uterus or in your breast. That's what oxytocin does, stimulates the contraction of your uterus during pregnancy or delivery. So this molecule here will cause the effect because in the membrane of the targeted cells, there are proteins referred to a receptor which are specific for these molecules. Specific means that this receptor will recognize only this molecule but not other molecules. When this happens, these will get activated and then will lead to transfer of the signaling from the outside to the inside. And then these start the transduction and then ultimately the response inside the cell. When it comes to sorry, these are going to be too fast. So moving on here. So the receptor here, usually they consist of three main domains. So they have three regions, three domains. You remember proteins domains? So they would have all a transmembrane domain, a domain that allows them to embed inside the cell membrane. They have to be anchored there. Then they would have an extracellular domain, which is also the signal binding domain. This is where the specificity comes. That's where the protein specifically can bind to a specific signaling molecule. And then they would have an intracellular domain here, which usually is the effector, the enzyme domain, or a kinase domain, which basically has an enzymatic activity. So the way they work is the binding of the signaling molecule through the domain causes a conformational change to the overall receptor, going through the transmembrane domain all the way to the intracellular domain. And then this leads to the activation of the enzymatic domain on the inside of the cell. This domain usually is a kinase enzymes. We'll tell you in a second. And then, so this is just an overview. And so what the kinase does, this intracellular domain, this is the process what we're talking about here, the process of reception, leads to the activation of these enzyme domain inside of the receptor, which is a kinase. What kinases do, what they do, they activate other molecules, in this case, other enzymes, by adding a phosphate group to them. So basically what they do, you can see here on the right, they take the specific molecule, target molecule, in this case an enzyme, an ATP molecule, and they transfer one phosphate group from this ATP molecule to the target protein. By doing that, they basically convert the target protein from inactive to active. These in signal transduction, so that's the first step in signal transduction, this leads to the activation of another enzyme, which is a kinase as well. These are different type of kinases. They have different target molecules that recognize. But this kinase will do the same thing. Will do, as being a kinase, will phosphorylate another target, which is another kinase, which then phosphorylate another target. We'll see the importance of doing all these steps later on, how amplification is achieved here, which then activates another target and so on. And this goes all the way until the final target protein is reached. The effector protein is the one that ultimately will cause the execution of the meaning of the signal molecule here. Let's say this had the meaning of stimulating this cell to contract or to duplicate, for example, to replicate. Now, this target protein will finally bring the final cellular response, which is going to be the duplication. So this is the reception, first step. Then there's a transduction happening through a series of protein kinases being activated one at a time in a hierarchical way. And finally, this leads to the activation of the cellular response. Now, obviously, these signals has to last only until it's needed. You don't want to have vasopressin causing contraction in your uterus forever. That would be kind of painful, right? You want to have it only when you're about to deliver the baby. Once the baby is out, You don't need to have that contraction anymore. So this signal has to be turned on and turned off. How is turned off? It's turned off by turning off basically these effector kinases here, these protein kinases here, by doing the opposite reaction. So now, these protein kinases are being turned on by the previous protein kinase by adding a phosphate group. Now you have other enzymes inside the cell called phosphatases. They're specific for each of these kinases, which do turn them back into the inactive form by removing the phosphate group. So kinases and phosphatases, basically enzymes that do the opposite thing. Kinases, they add the phosphate group, turning the protein on. Phosphatases, they remove the phosphate group, they turn it off. Okay, and all of this is balanced inside so that you have your signal happening, and then this cascade is going to be turned off and be ready to get another signal if necessary. Okay, so now the last thing is in signal transduction. Why do we need all of these kinases be turned on one after the other? Why can't we simply have a receptor that turns on the target protein and causes the response? The reason is that by doing this, you can have actually an amplification of the signal. So you can have that even the activation of a single receptor can be amplified inside here into a huge, huge signal. How? By having each kinase activating more than one kinase. So imagine this, the first kinase is the one that's part of the receptor, right? Imagine this activates 1,000 kinases, first level. And now each of these kinases activates 1,000 kinases as well. So now you have that one receptor here has activated 100 of them, 100,000 of this molecule, and so on. So imagine this activates another 100. So you're getting to 1,000,000 kinases that are finally activated. And so you can have that a single event here can activate still millions of final target proteins, so amplifying the signal, the original signal. You don't need to have at this point billions of signal molecules happen here. You just need a few of them, all right? Which it's what you need when it comes, for example, to signal at a distance. Your hypothesis cannot make millions of oxytocin molecules or vasopressin molecules. They make a few of them. Some of them get diluted in your blood, and then only a few of them reach this receptor. Despite that, you get the amplification of the signal through this kinase cascade. That's referred to as the amplification cascade of signal transduction there. All right. So in summary, what did we talk about in this lecture? We talked about two main things. The most important part that we talked about was how do molecules go across the cell membrane, this phospholipid bilayer. And we talked about there are two types of movements. Some of them do not require energy. They're referred to as passive movements. And so the two that we talked about are diffusion, whether it's simple, so the molecules, small molecules, they move on their own, small gases, and so on, through the membrane, following their concentration gradient, so that will be simple diffusion. Or sometimes some molecules, they do the same thing, but they need to be helped by passive transporter, so proteins. In that case, you will refer to as facilitated diffusion. When molecules cannot move across the cell membrane, you might still have water doing similar movement, which doesn't require energy. In this case, referred to as osmosis. In this case, the movement will go against the concentration gradient of the solutes. Then we talked about the second type, active movements type of type. So we have transporter that can move one molecule at a time against, in this case, of the concentration gradient. This will be referred to as active transport, which can be of two types, primary or secondary, depending on whether the ATP, the energy comes from binding directly to the ATP molecule or from dissipating the concentration of a second molecule, same direction or opposite direction. So primary or secondary active transport. In some cases, again, we talked about how instead of moving one molecule at a time, you can move large amount of molecules or large object through the production of vesicles. So we talked about that of exocytosis, endocytosis, whether it's pinocytosis, receptor-mediated, endocytosis and phagocytosis. Active transport and vesicle-mediated transport, they all require the energy in order to take place, and therefore they are referred to as active type of movements. And finally, spend the last few slides talking about how signal is transferred across the cell membrane through the 3 steps of reception, through a specific protein which binds specifically to a signal molecule. This causes transduction of the signal ultimately from the outside to the inside, and the amplification inside the cell until the final response is achieved.